Dear student, we will continue with the anatomy of flowering plant. Now we have to discuss the structure of transverse action of root and stem. So if you look at the root and compare the dicot root transverse action with monocot, you will see they both have epidermis, then they have the layer of cortical cells called cortex, and then the layer of endodermal cells. Inner to that is pericycle. Now, the region which is inner to the endodermis is whole known as stele, which consists of pericycle, vascular tissues, pith, and all these things. So, endodermis and after that pericycle, then you will see the vascular bundles are different in dicot and monocot. The number are different in dicot 2 to 4, whereas in monocot 6, more than 6. Okay, and you will see the size of pith, it is very prominent in dicot, whereas in monocot it is very small. And then there are certain tissues which are present between the vascular bundle, they are called conjunctive tissues, and pith is located at the center. So this is what is you will see in the transverse section. Similarly, if you see or compare the monocot stem and dicot stem, you'll find that in monocot stem the and in dicot stem both the epidermal cells are outermost, inner to that is the hypodermal cells, and in monocot it is single layer, whereas in dicot it is multi-layer. And then in the monocot stem you will not see the cortex tissues arranged in the different manner in the external and internal, but these tissues are called ground tissues in which the vascular bundles are scattered. The vascular bundle in both the cases are conjoint collateral, but they are closed in monocot and they are open in dicot. And you will find that the vascular bundle in stem, they are smaller toward periphery, and toward the center they are big, and in one bundle xylem and phloem both are present. So here the condition is endar, the protophlorium is located toward the inside, whereas the metaphlorium toward the outside. But if you compare it with the root, the condition is exarch, the protophlorium is out and the metaphlorium is toward the center. So here you will see um, medullary rays which are there between the vascular bundle, and they have a great role to play in the secondary thickening. So this is how you can compare all four. And dicot, the stem, and dicot root, there occur the secondary thickening. So next we will discuss that how the secondary thickening occurs. So if you see the next part is the secondary growth in the plant. So in the dicot stem, first we will discuss the dicot stems. They exhibit both, even the root, the primary growth and secondary growth. Primary growth occurs due to the cell division at the tip of root stem causing the elongation, whereas the secondary growth occur due to cell division in vascular cambium and give rise to increase in girth. So secondary growth occurs due to lateral meristem. Now there are two meristematic cells, vascular cambium and core cambium, and secondary growth occurs because of these cambial activity. First discuss the vascular cambium. It adds secondary tissues, the vascular region, that is it adds secondary phloem and secondary xylem. And vascular cambium is present between the xylem and phloem, and this is called intravascular cambium, and acquired the meristematic activity at a particular time. So the cambium is already present between the xylem and phloem, but at a specific time it acquires the meristematic activity and it will start dividing. Okay, so as a result, the intravascular cambium developed between the neighboring vascular bundle. So, if you look at the vascular bundle, cortex region is there, then medullary rays are there, and vascular bundles are there. Okay, so once it acquires the meristematic activity, what will happen? The cambium ring will form. Now, it is not only that meristematic of activity of cambium, but also the meristematic activity is acquired by the medullary rays which are present between the vascular bundles. So, as a result, what? The cambium is also present between the vascular bundle. Okay, this is called interfascicular cambium. Here you can see interfascicular cambium. The vascular cambium, and which is called intravascular, and the interfascicular cambium together form a complete cambial ring. If you see the vascular cambium, they have two different types of cell initiating: fusiform initial and ray initial. Fusiform initial, they are narrow elongated cell that give rise to secondary xylem and phloem, whereas ray initial, they give rise to medullary rays. Now, if you look at the cork cambium, here we have discussed the vascular cambium, which give rise to secondary phloem and xylem, and also you can say the ray initials, which present give rise to the medullary rays. Okay, cork cambium adds secondary tissue in the region of cortex. Okay, and it forms the cork and secondary cortex. So next step is the cambial activity we have to discuss. Now, once the cambium ring is formed, it is very active, means it acquired the meristematic activity and start producing the cells toward the inner and outer side. The inner cells add secondary xylem toward inside, and the outer one adds secondary phloem to the outside. The inner cell activity is more toward the inside, so more xylem cells are added as compared to secondary phloem. Therefore, xylem increases in bulk and the primary xylem is pushed toward the center and become non-functional but remain intact, and the pith get reduced. So if you see secondary phloem, the primary and secondary phloem get crushed due to the accumulation of secondary xylems because the activity is more toward the inner side. Further, the endodermis, cortex, epidermis are also ruptured due to the pressure exerted by the secondary xylem. So if you look at here, you can see primary xylem is there, and then it forms next layer is secondary xylem, then cambium ring is there, and you can say toward outside it adds secondary phloem and primary phloem is pushed out. So since the secondary xylem keep increasing, the outer things they move little out, and the the cortex endodermis, they get not endodermis, the cortex and the other cells, epidermal cells, they get crushed. Okay, so finally you will see that because of the activity of cork cambium, which is developed from the cortex. Okay, so the parenchyma and collenchyma cells of cortex undergo dedifferentiation and acquire meristematic activity to form cork cambium, which is known as phallogen cells. The cork cambium produces outer cells called cork and inner called cortex or phallodum. So here you can see outside the cortex, the secondary cortex is there, and it produces outer and inner layer. The outer one is called cork and the inner one is called secondary cortex, and in between is the cork cambium. Okay, so this is how the secondary growth occurs. Again, more activity is toward the outer side as compared to the inner side, so more cork you will find in the secondary during the secondary thickening. Now, beside inner and outer, you will find that some cell continue to grow radially, especially the ray initials, and forms the secondary medullary rays. So you will also see the secondary medullary rays. okay, which help in the conduction and transportation of water and mineral. Okay, so since the xylem is also known as wood, it is of two types. Before that, I'll just discuss the paridum and secondary phloem forms the bark. Secondary phloem is the living structure, whereas the paridum is the cork, and all these are dead structure. Okay, so bark consists of living and non-living structure. Now, secondary xylem is known as wood. It is of two types, soft and hard. Soft wood mainly consists of 90% of tracheids and is present in gymnosperm, whereas hard wood has no tracheids and they are present in the angiosperm. Now, depending on the vascular cambium activity, it is also known as spring wood and autumn wood. Spring wood is called early wood because it forms early, and autumn wood is called late wood. See, they together form one annual ring. Spring wood, the cambium here, the cambium activity is rapid. Okay, so this forms is in lighter color and has low density, and the vessels which are there in the wood, they have large cavities. But if you compare it with the autumn wood, the activity, cambium activity is less, you can say, not very active, and it is darker in color because it is slowly forming, and it will have high density, and it is the vessels are fewer in number. So this is the difference between then also the wood is categorized as the hardwood and sapwood. The hardwood, if you see the trunk portion of the tree, the transverse section, the darker region in the center is called hardwood, because it consists of many secretory product like tannin, oil, gums, and has hard, more durable and resistance to microbe, and it is a dead wood. Whereas the sapwood is the outer region of the stem. If you see the trunk piece, and there is no secretory product, it is light and it conduct water and mineral and also susceptible to microbes, and it is the living part. Okay, now we'll just discuss the secondary thickening in root. Just the same cells, vascular cambium and cork cambium, just like stem, they are responsible. But here you must know the structure of dicot root. Just to recall, you can see the vascular bundles are there, pericycle is there, and phloem is there. So here we will see how it takes place. The vascular cambium originate from the tissue located just below phloem bundle, that is here. Okay, just below the phloem bundle, that is this, and the this portion, pericycle tissue, and they become meristematic. So these two places become meristematic, you can see, and started here the meristematic cells started dividing. So wavy cambial ring is formed. So this is how it is formed. This become active and produces inner and outer cell. Inner is secondary xylem, outer is secondary phloem, and activity is more toward the inside. So you will find that secondary xylem growth is more toward inside as compared to the secondary phloem. Now slowly the wavy ring changes to the circular. So here you can see circular cambium. This is circular cambium. It pushes the primary phloem outward. Continuous activity crushes endodermis, cortex, and epidermis, and the cork cambium is formed. So dedifferentiation of cells in the pericycle produces cork cambium, and this cork cambium produces cork outside and secondary cortex inside. So cork plus cork cambium and secondary cortex is called peridum, and all the dead tissues are replaced by bark, and the bark has living and non-living tissues. So this is how the secondary thickening in the root occurs. So the difference basic is here the pericycle cells are involved, whereas in the stem the cortex cells are involved to form the peridum. Now, dedifferentiation and redifferentiation, just to understand, dedifferentiation is the formation of interfascicular cambium and cork cambium from fully differentiated parenchyma cells. Redifferentiation is a dedifferentiated plant cells once again loses its capacity to divide and become mature. So in the plant growth you will see differentiation, and again you will see dedifferentiation and redifferentiation. So this is all about the plant growth and anatomy of flowering plant. Thank you.